To prepare the grid box in AutoDock Tools, click on Grid, then select Macromolecule, and click on Open. Now, browse to your working folder and select the target protein structure in .pdb format. Once the protein is loaded, AutoDock Tools will automatically assign charges and prepare the macromolecule for docking. After the preparation is complete, save the processed protein file in pdbqt format. While saving, make sure to manually type the file extension, pdbqt, at the end of the file name before clicking Save. Save the file inside your working folder. Your target protein is now ready for grid box generation in molecular docking analysis. After saving the target protein in pdbqt format, click on Edit, then select Delete, and remove all molecules from the workspace. Now, load the prepared macromolecule again for grid box generation. Click on Grid, then select Macromolecule, and click on Open. Browse to your working folder and select the saved pdbqt file of the target protein. When the confirmation window appears, click Yes to continue. Next, go to Grid again and select Grid Box Preparation Settings. Click on Grid, then choose Set Map Types. Now, open the prepared ligand file in pdbqt format by selecting the ligand pdbqt file from your working folder. This step allows AutoDock Tools to identify the atom types present in the ligand for proper grid map generation. The ligand will now appear in the workspace along with the target protein. You can click on the ribbon structure of the protein to clearly visualize the position where the ligand is bound within the active site. This visualization provides an important clue for defining the grid box dimensions and positioning the grid correctly around the active binding pocket for molecular docking studies. Click on Grid, then select Grid Box. A grid box will now appear around the target region of the protein. You can adjust the X, Y, and Z coordinates to position the grid box over the active binding site. Next, adjust the size of the grid box by increasing or decreasing the number of points along the X, Y, and Z axes. Make sure that the grid box is sufficiently large to completely cover the active site and provide adequate space for the ligand to rotate, move, and explore different binding conformations during the docking simulation. After setting the number of points in the X, Y, and Z dimensions and properly centering the grid box along the X, Y, and Z coordinates, review the grid box placement carefully around the active site. Once the grid box parameters are finalized, click on File, then select Close Saving Current to save the current grid box configuration. Next, click on Grid and select Output. Save the grid parameter file in .gpf format inside your working folder. While saving, make sure to type the file extension, .gpf, at the end of the file name before clicking Save. The .gpf file contains all the grid box parameters required for running AutoGrid during the molecular docking process. You can also open the saved .gpf file using Notepad or any text editor to verify the written grid parameters. Inside the file, you can check important docking parameters such as the grid box dimensions, center coordinates, spacing, receptor file name, ligand atom types, and other AutoGrid settings used for molecular docking analysis. Now, we will prepare the docking parameter file for molecular docking. Click on Docking, then select Macromolecule, and choose Set Rigid File Name. Browse to your working folder and open the prepared target protein file in pdbqt format. Next, click on Docking, then select Ligand. A new window will appear displaying the available ligand files. From this window, select the prepared ligand file. After selecting the ligand, a separate window will open showing the ligand properties and docking parameters. Review the information, then click on Accept to continue. Click on Docking, then select Search Parameters, and choose Genetic Algorithm. A parameter window will appear where you can set the GA run value. You may increase or decrease the number of GA runs depending on your docking requirements. The selected number of GA runs determines how many docking conformations or binding poses will be generated during the molecular docking simulation. Generally, a higher number of GA runs increases the probability of identifying the most stable binding conformation of the ligand with the target protein. For this tutorial, keep all other parameters of the genetic algorithm at their default settings. These default parameters are generally optimized for standard molecular docking studies in AutoDock. Next, click on Docking, then select Docking Parameters. A parameter window will appear and click on Accept to continue. Next, click on Docking, then select Output. Choose Lamarckian genetic algorithm as the docking method. Now, Save the docking parameter file in .dpf format inside your working directory. While saving, make sure to type the file extension, .dpf, at the end of the file name before clicking Save. The .dpf file contains all the docking parameters required for running the AutoDock simulation. Now, your working folder contains both the .gpf file and the .dpf file required for molecular docking. You can open the saved .dpf file using Notepad or any text editor to verify all the written docking parameters. Inside the file, you can check important details such as the receptor and ligand file names, grid box settings, genetic algorithm parameters, number of GA runs, docking method, and other simulation parameters used during the AutoDock docking process. Before running AutoGrid and AutoDock, make sure that your working folder contains both the AutoGrid.x and AutoDock.x executable files. To run AutoGrid, click on Run, then select Run AutoGrid. A new window will appear. In this window, select the AutoGrid.x file in the Program Pathname section. Select the prepared.gpf file in the Parameter Filename section. After selecting the required files, click on Launch to start the AutoGrid process. A command window will appear on the screen showing the grid map generation process. Wait until the window automatically disappears, indicating that the AutoGrid run has completed successfully. After the successful completion of AutoGrid, the .gpf file will be processed and converted into a .glg log file. You will also notice that several new grid parameter and map files are generated automatically inside your working folder. After completing the AutoGrid process, we will now run AutoDock for molecular docking simulation. Click on Run, then select Run AutoDock. A new window will appear. In this window, select the AutoDock.x file in the Program Pathname section. Select the prepared.dpf file in the Parameter Filename section. After selecting the required files, click on Launch to start the AutoDock simulation. A command window will appear showing the docking process and conformational search performed by AutoDock. Wait until the window automatically closes, indicating that the docking run has completed successfully. After the successful completion of the AutoDock run, the .dpf file will be converted into a .dlg.